Bismillah Rahman Rahim. Assalamu alaikum. Today I welcome all my students of second year MBBS and BDS to the regulation of pancreatic secretion. This is just the continuation of the previous lecture in which we have done the secretions of pancreas. Now, what are the basic stimuli that cause the pancreatic secretion are very important. First one is acetylcholine, then cholecystokinin, secretin. Now, acetylcholine is released from the parasympathetic vagus nerve endings and from other cholinergic nerves in the enteric nervous system. Cholecystokinin is secreted by the duodenal and upper duodenal mucosa when food enters the small intestine. Secretin is also secreted by the duodenal and jejunal mucosa when a highly acidic food enters the small intestine. Now, the first two of the stimuli, acetylcholine, cholecystokinin, they stimulate the acinar cells of the pancreas, which can lead to the production of large quantities of pancreatic digestive enzymes, but relatively small quantities of water and electrolytes to go with the enzymes. Secretin, in contrast to the first two basic stimuli, stimulates secretion of large quantities of water and solution of sodium bicarbonate by the pancreatic ductal epithelium. Multiplicative effects of different stimuli. When all the different stimuli of pancreatic secretion occur at once, the total secretion is far greater than the sum of secretions caused by each one separately. The various stimuli are said to multiply, or they are going to augment, or they are going to potentiate, or they are going to accelerate one another. Thus, the pancreatic secretion normally results from the combined effects of the multiple basic stimuli, not from one alone. Now, there are different phases of pancreatic secretion, which are cephalic and gastric phases. During the cephalic phase of pancreatic secretion, the same nervous signals from the brain that cause secretion in the stomach also cause acetylcholine release by the vagal nerve endings in the pancreas. This causes moderate amounts of enzymes to be secreted into the pancreatic acini, accounting for about 20% of the total secretion of pancreatic enzymes after a meal. But little of the secretion flows immediately through the pancreatic ducts into the intestine because only small amounts of water and electrolytes are secreted along with the enzymes. During the gastric phase, the nervous stimulation of enzyme secretion continues, accounting for another 5 to 10% of pancreatic enzymes secreted after a meal. But again, only small amounts reach the duodenum because of continued lack of significant fluid secretion. Then we have the intestinal phase. After chyme leaves the stomach and enters the small intestine, the pancreatic secretion becomes copoish. Copoish means enough, mainly in response to hormone secreted. Secretin is a polypeptide containing 27 amino acids having the molecular weight of 3400. They are present in an inactive form, prosecretin, in so-called S cells in the mucosa of duodenum and jejunum. Secretin stimulates copoish secretion of bicarbonate ions, which neutralizes acidic stomach chyme. When acid chyme with pH less than 4.5 to 5 enters the duodenum from the stomach, it causes duodenal mucosal release and activation of secretin, which is then absorbed into the blood. The one truly potent constituent of chyme that causes the secretin release is the hydrochloric acid from the stomach. C. Hydrochloric acid plus sodium bicarbonate can lead to formation of sodium chloride and carbonic acid. Secretin in turn causes the pancreas to secrete large quantities of fluid having high concentration of bicarbonate ion up to 145 milliequivalents per liter, but a low concentration of chloride ion. The secretin mechanism is especially important for two reasons. Why? Because first, secretin begins to be released from the mucosa of small intestine when the pH of the duodenal contents falls below 4.5 to 5, and its release increases greatly as the pH falls to 3. This immediately causes copious secretion of pancreatic juice containing abundant amounts of sodium bicarbonate, and the net result is then following reaction in the duodenum. Then the carbonic acid immediately dissociates into carbon dioxide and water. The carbon dioxide is absorbed into the blood and expired through the lungs, thus leaving a neutral solution of sodium chloride in the duodenum. In this way, the acid contents emptied into the duodenum from the stomach becomes neutralized. So further peptic digestive activity by the gastric juices in the duodenum is immediately blocked. Because the mucosa of the small intestine cannot withstand the digestive actions of acid gastric juice, this is an essential protective mechanism to prevent development of duodenal ulcers. Bicarbonate ion secretion by the pancreas provides an appropriate pH for action of the pancreatic digestive enzymes, which function optimally in a slightly alkaline or neutral medium at a pH of 7 to 8. The pH of the sodium bicarbonate secretion averages 8. Cholecystokinin, its contribution to control of digestive enzyme secretion by the pancreas. The presence of food in the upper small intestine also causes a second hormone, cholecystokinin, a polypeptide containing 33 amino acids to be released from the eye cells in the mucosa of the duodenum and upper jejunum. This release of cholecystokinin results especially from the presence of proteases in peptones and long chain fatty acids in the chyme coming from the stomach. Cholecystokinin, like secretin, passes by way of the blood to the pancreas, but instead of causing sodium bicarbonate secretion, causes mainly secretion of still much more pancreatic digestive enzymes by the acinar cells. The differences between the pancreatic stimulatory actions or effects of secretin and cholecystokinin demonstrate that there is intense sodium bicarbonate secretion in response to acid in the duodenum stimulated by secretin, a dual effect in response to soap fat, intense digestive enzyme secretion when peptones are stimulated by cholecystokinin. See? Now look at this diagram. This is going to give us the idea what is the role of secretin, cholecystokinin, vagal stimulation, that is acetylcholine, then acid from the stomach. Now see what happens is that the acid from the stomach releases secretin from the wall of duodenum, fats and amino acid. They cause the release of cholecystokinin. Then vagal stimulation releases the enzymes into acini, and then the secretin causes the copious secretion of pancreatic fluid, the bicarbonate ion, the cholecystokinin causes secretion of enzyme. So this is all about the control of pancreatic secretions, how they are being controlled by the nerve vagus and by different enzymes, how hormones are being controlling the enzyme release, how. So this is all about today's lecture, and give a good reading to this topic. Till then, stay blessed and Allah Hafiz.